You did the diet. Again. Lost some weight, felt good for a few weeks, then watched the belly slowly reappear like nothing happened. And somewhere in that cycle, a quiet voice started saying, "This was your fault. Maybe your portions crept up. Maybe you weren't consistent enough. Maybe your metabolism is just broken." That story isn't accurate, and the research backs that up. For a large number of people over 40, visceral fat, the kind packed around your abdominal organs, doesn't return because of a lack of effort. It returns because of a biological loop anchored in the liver. A loop that runs in the background whether you're dieting or not. If you've tried low carb, low fat, or both. If you've lost weight and watched it land back in the same spot. If you're doing everything suggested but your waist isn't responding the way the rest of your body does, this is for you. What follows isn't a supplement pitch. It's the published metabolic science of why visceral fat is self-sustaining, and what the research actually says you can do about it. Act 2 is where the loop lives, inside your liver. Act 3 is what the liver ships back out, and where it lands. There's a detail at the end of Act 2 that most health content never reaches. Stay with me. Your liver sits at the center of a one-way delivery system most people have never thought about. When you eat carbohydrates or sugars, what's absorbed through your gut doesn't drift freely toward the liver eventually. It gets collected by a large blood vessel called the portal vein and delivered directly to the liver before it reaches anywhere else in your body. Your muscles don't get first access. Your fat cells don't. Your liver does. Now here's where it gets specific. Fructose, the sugar in fruit juice, agave, high fructose corn syrup, and most sweetened processed foods, travels this same private route. Research published in Digestive Diseases and Sciences confirmed that fructose arrives at the liver through the portal vein at far higher concentrations than it reaches any other tissue. The liver intercepts nearly all of it. And once it arrives, fructose doesn't behave the way glucose does. Most sugars need insulin to enter cells and get processed. Fructose doesn't. It bypasses the usual checkpoint, enters the liver cell, and gets metabolized fast, in large amounts, with no built-in governor on the process. The liver now has a large load of raw material and has to decide what to do with it. If your energy stores are depleted from exercise, some gets used appropriately. But for most people in a normal day, the liver's stores are already reasonably full. So it activates a secondary program. It starts making fat. This process is called hepatic de novo lipogenesis, the liver manufacturing brand new fat from scratch, using sugar as raw material. Think of it as a factory inside the liver that converts excess carbohydrate into triglycerides ready for export. Two molecular switches control this factory. SREBP1C, activated by insulin, and ChREBP, activated directly by glucose and fructose, independently of insulin. Research published in Genes and Nutrition identified these as the primary transcriptional regulators of the liver's fat production program. When both switches are on together, the factory runs at full capacity. How significant is this in practice? Research published in Digestive Diseases and Sciences used isotope tracer studies to compare people with normal liver fat against those with elevated liver fat. In healthy individuals, de novo lipogenesis contributed around 10% of liver triglycerides. In those with excess liver fat, that figure had climbed above 23%. Separate controlled studies found that 3 weeks of carbohydrate overfeeding increased liver fat by up to 27%, while total body weight rose by only 2%. The liver was filling faster than the scale would ever reveal. Before we get to where the liver ships its newly made fat, one thing is worth understanding, because it explains why reducing fructose specifically makes such a difference. When glucose arrives at the liver, the body has a built-in throttle. An enzyme called phosphofructokinase slows glucose processing when energy stores are already full. Fructose bypasses this throttle entirely. Fructose enters via a different enzyme, ketohexokinase, that has no feedback inhibition. No governor. When fructose comes in, it gets phosphorylated immediately, the liver's ATP drops rapidly, and the fat-making cascade fires. Research published in Frontiers in Pharmacology described this as a key reason fructose drives de novo lipogenesis more aggressively than high-fat diets at equivalent calorie loads. Insulin resistance adds another layer. As insulin sensitivity declines, which happens gradually through your 40s, insulin levels run higher in response to the same carbohydrate loads that caused no issue at 30. Higher chronic insulin means the SREBP1C switch stays on longer. The factory gets a prolonged green light. And because fructose doesn't require insulin for its own metabolism, this double activation, insulin driving SREBP1C and fructose driving ChREBP, continues even as insulin resistance worsens. Research published in Digestive Diseases and Sciences made this explicit. Fructose can stimulate the fat-making program even in settings of significant insulin resistance. The factory doesn't slow when the insulin signaling system starts to fail. Your liver is making fat, steadily, independently, without you necessarily overeating. Now the question is, where does it go? Your liver doesn't store the fat it makes. Instead, it packages newly manufactured triglycerides into structures called VLDL particles, very low-density lipoproteins. Think of VLDL as the liver's delivery fleet, trucks loaded with fresh fat, dispatched into the bloodstream, sent out to tissues around the body. Research published in Arteriosclerosis, Thrombosis, and Vascular Biology confirmed that insulin resistance, increasingly common after 40, is directly linked to overproduction of these large VLDL particles. The fleet runs more trips. Each truck carries a heavier load. At the capillaries of various tissues, an enzyme called lipoprotein lipase, LPL, acts as a dock worker, freeing the fatty acids for uptake by surrounding cells. A 2021 review in Metabolism, Clinical and Experimental, describes how these LPL docking stations exist at the capillaries of adipose tissue, skeletal muscle, and the heart. Visceral adipose tissue, the fat packed around your liver, stomach, and intestines, is one of the most LPL-active depots in the body. When the liver's trucks cruise through abdominal circulation, the visceral depot waves them in and reliably offloads the cargo. And now, the loop closes. When you diet and visceral fat cells release stored fatty acids, they drain directly into the portal vein, the same route nutrients take from gut to liver. Those fatty acids go straight back to the liver, get repackaged into new VLDL trucks, and a portion ends up back at the visceral depot. You diet, visceral fat releases, the liver reprocesses. VLDL trucks redeploy. Visceral fat reaccumulates. Research published in the American Journal of Physiology documented that people with elevated liver fat maintained significantly higher de novo lipogenesis rates, even when matched with others eating identical diets. The loop sustains itself through the metabolic state the liver is already in, not just what you ate today. The scale moves. The loop doesn't stop. Understanding the loop is half the battle. What do peer-reviewed studies actually suggest can interrupt it? The first lever is reducing liquid fructose. Not fruit. Whole fruit delivers fructose bound to fiber, which slows portal delivery and changes how the liver handles it. The concern is specifically fructose in beverages and processed foods, sodas, juices, syrups, sweetened packaged products. Research published in Nutrients and Frontiers in Pharmacology consistently shows that reducing this category lowers the rate at which the liver's fat factory runs. One study in adolescents with confirmed liver fat showed that dropping free sugar intake to around 1% of daily calories, compared to a typical 10%, produced significantly greater improvements in liver fat over 8 weeks. The second lever is carbohydrate quality. Not low carb as an ideology, but recognizing which carbohydrates drive the ChREBP and SREBP 1C switches hardest. Refined, rapidly digested carbohydrates produce a far stronger fat-making signal than the same calories consumed alongside fiber, protein, and fat. This doesn't require calorie counting. It requires attention to how fast what you're eating hits your bloodstream. The third lever is consistent movement. Research published in the Journal of Hepatology found that regular physical activity reduces liver fat independently of weight loss, by increasing the rate at which the liver burns incoming fatty acids for fuel rather than packaging them onto VLDL trucks. Even moderate walking shows measurable effects in intervention trials. The fourth lever is protecting muscle mass. Skeletal muscle is another major LPL docking station. More active muscle means more of what the liver ships gets directed toward energy rather than storage, which is why adequate protein during a caloric deficit consistently produces better visceral fat outcomes than calorie restriction alone. None of these changes are instant. Liver fat can begin declining within weeks of meaningful dietary change, but the full recalibration takes months of consistent effort. Your body isn't against you. It's running an ancient program in a food environment it was never designed for. This loop doesn't run at the same intensity in everyone, and that's not a cop-out. It's biology. Research published in Nature Metabolism identified variants in 2 genes, PNPLA3 and TM6SF2, that make some people significantly more prone to hepatic fat accumulation on diets that cause no such problem in others. If you've watched a friend eat the same things and carry far less belly fat, this is one real reason. It's not perception. It's gene expression. Age matters too, independently of diet. After 40, insulin sensitivity tends to decline steadily. The same carbohydrate load your liver handled cleanly at 28 now triggers a longer SREBP1C activation. More factory time, more VLDL trucks, a depot that fills more readily. That's not a willpower failure. It's a shift in how the liver reads hormonal signals. For women, menopause adds a specific mechanism on top of this. Estrogen directly influences hepatic lipid metabolism, and its decline accelerates liver fat accumulation in ways that don't respond to the same interventions that worked at 35. If you're over 40, have visceral fat that appeared gradually, see elevated triglycerides on a blood panel, or have been flagged for borderline liver enzymes, these aren't disconnected findings. They're the same loop, measured from different angles. Visceral fat isn't stubborn because you're doing something wrong. It's persistent because a biological loop keeps resupplying it. The portal vein delivers fructose and refined carbohydrates to the liver first. ChREBP and SREBP1C fire up the fat manufacturing program. Triglycerides load onto VLDL trucks and ship into the bloodstream. The visceral depot, a high-capacity LPL dock, accepts the delivery. When you diet, visceral fat drains back through the portal vein to the liver. The cycle continues. Interrupting it means going upstream, reducing liquid fructose, improving carbohydrate quality, staying consistently active, and protecting lean muscle mass. None of that requires perfection. All of it requires understanding what's actually happening. And that's what you now have. If this changed how you think about your metabolism, share it with someone who's been stuck in the same frustrating pattern. Drop a comment telling me which part of this landed hardest for you. That exchange genuinely helps others. And for more content that takes the published science seriously, without wrapping it in hype, subscribe. There's more of this coming.